Okay, so this revision video is on the primary and secondary immune response and being able to draw a graph of those responses. So the first thing I want you to do as a revision task here is to label the axis and then draw the line to show how the antibody concentration changes in the primary and secondary immune response. And then the second task I want you to do is try and explain it in terms of these graphs, in terms of the cells involved, so your knowledge of the non-specific and specific immune responses in the cells involved. And you can either do this by writing it out, you can draw it, or you can describe it out loud to yourself, or you could describe it to a sibling or a parent. I'm sure they'd appreciate that. Okay, so pause now and do those tasks, and then we'll go through it. Okay, so let's look at what you should have got. So on your y-axis, for the labels, you should have had antibody concentration in the blood, and then on the x-axis, you should have had time, and then your graph for the primary immune response is that it shouldn't start from the origin. You should start, there's a bit of a lag, and we'll go through why that is, and you should understand that. But you'll start to see an increase in antibody concentration. It'll reach a peak, and then it will drop. There shouldn't be really a bump on there, but it will drop down. So it's a peak like that. And that peak can be sort of 2 weeks, 21 days. That lag phase can be maybe 7 days before antibodies have been produced. And then your secondary immune response, you don't have such a lag. It's very quick. So the lag phase is much shorter. The increase in antibody concentration is much more rapid, and the concentration is much greater in the secondary immune response. So you should have a graph that looks, a line that looks like that. So your secondary immune response should be faster and greater. Okay, so let's consider the second question I asked you, which was relating this to what's happening on a cellular level. So let's first focus on this lag phase. So why is there a lag phase? Well, a few things have to happen, as we know. First thing, once the pathogen has entered the body, there will be phagocytosis by phagocytes of the non-specific immune system of the pathogen. They take it up, they destroy it, and then they present some of that antigen on their surface. They are then antigen-presenting cells. What happens next? Well, this is the connection between the non-specific and specific immune system. So helper T cells, then with a receptor that's specific for that antigen, will bind. What happens to those? We don't want to move that. What happens to those helper T cells? Well, they divide by mitosis. They produce lots of versions of themselves, and they can do one of four things, remember. We're going to concern ourselves with only one thing today, which is that they activate B cells. So let's just take a look at the B cell. Slightly blocked off there because of something else. Let's try and move that a bit that way. There we go. So a B cell will have or a B cell will have antibody on its surface that is specific for the antigen on the pathogen. It binds, takes it up by endocytosis, and then presents the antigen on its surface. One of these, or these helper T cells that have divided as a consequence of binding to the antigen on the antigen-presenting cell, bind, and in binding to that antigen on the surface of the B cell, activate the B cell. What does that do? Well, one of two things. We get production of antibodies. Where do we get the antibodies from? Well, the production of the plasma cells, and also the activated B cell will produce memory B cells. And remember, that process is clonal selection. So actually, all we've looked at there in that phase is, if we look at this lag phase, that counts for that part of what's going on, and in fact, sort of up to this part. So all of this here has happened within that lag phase. This has happened before we even get antibody production. And then obviously plasma cells are produced, so they start producing those antibodies, and we see this increase in antibody production until the pathogen is hopefully destroyed. Remember, that happens by agglutination and opsonization, which is that process by antibodies binding to pathogen and enhancing phagocytosis. Okay, so that's the primary immune response. Remember that there will be many of these memory cells, if they can stay on the board here. So many of these memory B cells produced. So from having maybe a few in your blood, you suddenly have a lot circulating your blood and tissue fluid. So next time you encounter that same pathogen, and I've drawn the arrow here, and I've put second infection with pathogen, and I've put antigen in brackets because obviously it's the antigen that's the key thing here. I've somehow converted that to red. I didn't want that. Let's put that back to blue. I wanted to draw a pathogen. That's what I want to do. So next time you encounter this pathogen, it's going to bind to these. It's more likely to encounter these memory B cells because there's lots of them already waiting in the blood, waiting for this pathogen or the antigen on it. The consequence of that is that they can very, very rapidly differentiate into plasma cells, lots of plasma cells, because there's going to be lots of these memory B cells around. So you're going to get lots of plasma cells produced very quickly, and as a consequence, lots of antibodies produced quickly. Hence why you get this very rapid increase in antibody production and this huge increase in antibody production because of these lots of memory B cells that can become lots of plasma cells and produce lots of antibodies. So that's the explanation of that graph. There we go.